to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 62nd episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for over 28 years. I'm here with my co-host Dominic Guarino.
Hi, my name is Dominic Guarino and I've been in the LBA and cell and gene therapy space for over 28 years now.
So Dominic and I are senior scientific advisors at KCS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCS's services. Today we're here with a couple of very special guests, John Bucksath, CEO of KCAS, and Marc Essodaigui, General Manager of Active Biomarkers in Lyon, France. I'm located in upstate New York, John and Dom are in Kansas, Marc is in France, and our producer is in Missouri, so we're truly all over the map. We're thrilled to have you listening to the 62nd episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we're looking forward to a fun episode today. To kick us off for podcast number 62, Dominic is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, as John mentioned, we have a jam-packed show today. We have two very special guests. I'll give them an opportunity to say hello. John, maybe you can go first.
Well, hi, Dom. Hi, John. Thanks for having me on the show. It's great to be here. I think this is my third time being on the podcast, so appreciate you guys having me on board.
And Marc, why don't you say hello real fast?
Bonjour. Thank you for your invitation. I'm very happy to be with you today.
Yeah, as I mentioned, we have two very special guests. And today's main portion, we're going to go over an introduction of active biomarkers. We'll go over a little history of active biomarkers. And then we'll really dig into the path to acquisition. And then what does it mean for KCS and Active, and the collaborations between our two great organizations, as well as what the acquisition means for our current customers and what it means for our future customers. Well, John, why don't we just jump right into the news? As always, we'll talk about news issues related to COVID. And this first one's a very interesting topic, or at least an interesting publication. It's related to repeat COVID infections. John, we've always said from the start of all this that getting COVID is not something you wanted to do. Like, maybe people did measles parties or chickenpox parties as kids. And that's definitely not the case here when it comes to COVID. And John, you can tell us a little bit more about this interesting study out of, I believe it was WashU, massive numbers of people they studied. And, we don't want to scare anybody out there, but John, why don't you tell us a little bit more about how you don't want to get COVID, like we've been telling you from the beginning.
Yeah, so basically the headline is repeat COVID infection deadlier than the first go-round, says study. And like you say, it's from the Washington University School of Medicine, St. Louis. That's struggling with my nephew's name.
WashU won't like you for that. No,
I know. The route to that is I've just come back from a christening in London where my great nephew was christened Louis, so I've got that in my head. So anyway, study done by the Washington University School of Medicine, St. Louis, where they actually looked at data on over 440,000 patients in Department of Veteran Affairs system from March 2020 to April 2022. Looked at 41,000 individuals with 2 or more infections and 5.3 million non-infected individuals. And from looking through the data, their overall conclusion was that people who have recurrent instances of COVID actually have more severe instances of COVID overall. They were concerned going into it that people sort of say, if I'm infected, then it's not going to have any impact on me if I get infected again. And they actually found that the data said the exact opposite. And so their recommendations is continue to really be careful about avoiding infection because like I say, the chances are that if you get infected the second or third time, you're going to have worse symptoms than you had before.
Yeah, it's an interesting study. It's very large, as you mentioned. The sample size is huge. I think it just boils down to what we had kind of said originally. COVID is not something that you want, and
Mhm.
the idea of getting it a second and third time would have thought maybe there was a chance, but to me there seems like there's something, the immunologist in me's kicking in a little bit, and there's got to be something underlying with the segment that is more susceptible to hospitalization and even potentially death with repeat infections. As I've always sort of preached, I think there's something about the receptor that it goes through, the ACE2 receptor there. I wonder, a lot we learned on HIV, there was CXC receptor mutations. I still wonder if that's part of what's happening here, either the expression levels of them or potentially just, mutations that could protect you or make you more vulnerable. It just seems like, well, the stats are what they are, but it definitely seems like there's got to be plenty of people who've had infection a few times and are probably protected. But again, that's just me speculating and just kind of talking about it, right? It's definitely not what we would have expected, not what we would have hoped in terms of, if you got infected a few times, you would have thought you would have been
Sure.
somewhat protected. And that's not the case, which is why you get a booster shot and why you get a flu vaccine every year as well. So I think it's kind of hopefully falling into that category. And one upside is it is talking, this article does say worldwide deaths from COVID are down.
Oh yeah, absolutely. Yeah.
But influenza and RSV are up on the rise. And I'm pretty sure I had a respiratory virus infection last week. So
I think that's it. It's basically that is the current picture, and certainly cases are down, but then we're going into a phase where our habits change, where we've been outdoor, at least in the northern hemisphere, we've been outdoors. Now we'll be more indoors, and so who knows what impact that has on things. So just watch it. I think as we've advocated all along, is be careful. That's really it.
Yeah, you should still social distance. If you want to wear a mask, wear one. I mean, I've always, long before COVID came, I was always like, try to stay arm distance away from me because I'm a little bit of a germaphobe to begin with. And so I think those practices just should be maintained. And, but life goes on. And I do think you're seeing more and more kids in school, and certainly just in general, we're going to get sick, but not with COVID. And so I hope we don't overreact in terms of, the measures we take. But anyhow, John, I
was in London prior to this. I was just last week, and obviously I was out and doing Christmas shopping, and shops were packed. But life was almost returned to normal there. You see a few people in masks, but it really was a much more low key thing than it has been before. So my take on all this is just be cautious and
wash your hands.
Sorry?
Wash your hands, everybody.
Yeah.
Right? Simple stuff.
But I traveled on the underground, not excessively packed underground, but I didn't have any issues. But then I'm vaccinated all the way through the bivalent boosters, so I think that obviously I've got some degree of protection. But I just thought this was a article worth highlighting that the reinfection piece is actually. Is something you still want to avoid. Don't think because you've been infected once that you're really in good shape if you get infected a second time. And I know we're dealing with family members who are on like the second or third infection, and it's obviously not a good place to be. Let's move on to the next topic.
Yeah.
This is a little bit more out there. It's still a COVID. Still a COVID topic, but I thought it was interesting because we have vaccines that obviously have had significant uptake, but if you look at the media, there's not really much following in terms of other treatments. We've had antibody treatments, but then the virus has mutated and rendered them ineffective. Shionogi have just had their antiviral approved in Japan, but not worldwide. So we don't really have many of those second or other alternatives to vaccination for treatment, etc. And this is very early work, but I just thought it was very, very cool kind of work, and may have implications obviously it does have implications for other viruses, where it's programmable molecular scissors to snip COVID causing virus. And basically, scientists from University of Cambridge have developed synthetic enzymes that could circumvent the challenges of dealing with viruses, where they're actually using the body to help themselves replicate, which gives little scope for targeting with antiviral drugs. So that's part of the challenge of antiviral drugs. So these synthetic enzymes are there to target genomes of viruses and cutting them into pieces and killing them. So it's like these programmable molecular scissors, as their creators described them, can be customized to deal with different viruses. And they published about this in Nature Communications, where they actually started working Ebola virus, but when COVID hit, then they adapted to the SARS-CoV-2 virus, and have shown proof of principle. That's really where it is at the moment. But I thought it was, as a potential future thing, this is just amazing.
Yeah, it sure is. I mean, like, just one of the collateral benefits of COVID is this type of technology, right? This dates back to 2014.
Yep.
They're using it on Ebola. They pivoted to COVID. And sure enough, they still have work to do, right? This is still in an early stage. They need to use electroporation to get it into the cell lines. But, this is like, I feel like it's like Star Trek type, right? Like these are things that are so targeted, and they still have to overcome how they're going to get it in, which I find fascinating. They talk about inhaling it, which has always been, I believe I've mentioned this in the past, when you think about how you get COVID is through inhalation, it would make sense to treat you as well as even vaccinate you. Might make more sense to go through the pathway that the virus goes. But this is really cool stuff in that they're using they're mimicking nature, but both synthetic DNA and RNA. They make these XNA zymes, which is very, very cool. I mean, it's a promising step forward. And as you said, there's still a very big need for therapeutics for COVID. So just a great article all around and really cool stuff.
So here's how it works. They work by recognizing and cutting sequences of nucleotides they're so precise that they won't cut RNA even if a single nucleotide differs from the programmed sequence. And because the platform's core structure remains the same regardless of what sequence is targeted, that's how they pivot from virus to virus. So when they started with Ebola, when they looked at it, SARS-CoV-2, they basically let's see where it is. Sorry, they basically went for the RNA sequence that related to SARS-CoV-2 virus and used that to attack it. Let's see.
Yeah, single nucleotide differences is what that's what it has to have that in order to between viruses or even self, right? But yeah, go ahead, John.
So if you want to talk about the lab piece, which is really interesting, they've tested them in artificial conditions, which is why we say this is all very, very early. Just to show that they were capable of cleaving the SARS-CoV-2 RNA, they went to cells incubated with live virus using a method called electroporation. They forced the XNA enzymes into bioluminescent cells that would glow when they came infected with SARS-CoV-2, then incubated them with the virus. The luminescence of the infected cells containing nanostructures was about 75% lower than those without them, and further studies measuring the amount of viral RNA showed that the XNA enzymes had inhibited infection. So it's really proof of principle stuff. It's cool. Is the best way I can put it. And then the next challenge is, how do we get it into the body to actually see how we can make this a practical solution to viruses, because this isn't just about COVID. This is about long term what other viruses might emerge.
Yeah, I mean, luminescence is something we use all the time at KCAS. We have luminescence methods for, they're cell-based methods, as you kind of articulated. You just load up a cell with something that will light up, luminescent, and then usually it comes from like algae, I think, is how the form of it, and then depending upon what your outcome is, you can look at either the cell dividing and losing it, and in this instance, what they did is exactly that. Electroporation is they send currents through a cell. It kind of causes the cell to get a little leaky and allows for the XNA zyme to enter the cell without destroying it. It literally kind of swells up and creates pores, electroporation, as the word indicates. So then through passive movements, it'll make its way into the cell. And that's where in this study, they showed that the 75% lowering just means it was able to cut the the virus out of them and subsequently kind of get an infected cell to be healthy. And so,
Sure. Yeah.
75% efficacy, John, we've talked about that too, in terms of we still don't know how many COVID particles are needed to cause infection and all that, but certainly lowering it by 75% would prevent it from aggressing, and more than likely your immune system would just clear it and you're not going to get sick. But it is good stuff. We can maybe move on to the next one, but you can see that you and I are really kind of geeky when it comes to this, because it is this is the next wave of stuff. I mean,
sure, absolutely. There's a few of these today are in that category, and like you said, what you talk about delivery. Well, now they're going to explore would it be inhaled, or do they go the lipid nanoparticle route, which plays into another topic, which there's so much focus on that at the moment. Yeah. So let's move on to non-COVID, and this is overeating sweet treats ? Scientists find what drives this desire. So basically, this is a team from Cold Spring Harbor Lab in Laurel Harbor, New York, have published in Nature Neuroscience, talking about that neurons in the part of the brain that control the fear response may also be responsible for the tendency to overeat fatty, sugary foods. Again, it's early work, but it has implications how you might treat this kind of thing in the long term. So they actually discovered that activating neurotensin neurons in the amygdala can regulate food preferences, feeding behavior, and metabolic health. And like I say, their findings could eventually form the basis for future weight loss therapies. I'll throw it back to you if you want to say something on this.
Yeah, I say, give me some of this is what I say to this. I definitely feel like there are times when I find myself munching on something thinking, why am I eating this?
It's your neurotensin.
It's literally why did I just spend 45 minutes on a treadmill to have this piece of chocolate, right? But there's always been powerful studies done on the brain. Neurons, especially with things like cocaine, they studied that pretty extensively in the 90s and 2000s. And this just falls right into that sort of category. I mean, your brain is a very powerful organ, and it's not surprising that they have identified areas of the brain that you can kind of try to knock it out or diminish. The one drawback here is the brain is such a mesh of networks that is still understudied and still evolving, that it just, I think it's great, and obviously there's a lot of work to be done on it, and they're looking at all sorts of pathways and so forth. But the redundancy of the brain is where I'm going, and I wonder, are you knocking something out, and could you, when I say knocking out, you're kind of diminishing the desire for sweets, but are you, and that's a metabolic pathway, you just wonder about the collateral piece.
Sure. Yeah, absolutely. So again, this is work we've done in mice. But they were actually looking at a structure within the amygdala called IPAC, or the interstitial nucleus of the posterior limb of the anterior commissure. So we'll stick with IPAC on that. So this is where the neurotensin comes in. Neurons in this region express neurotensin, a hormone that has been implicated in reward pathway signaling and is activated by stimulation of taste cells. And they basically did a number of treatments where they looked at how cells responded to a healthy versus unhealthy diet. So either giving regular food or high-fat food to two separate groups of mice, and found that the neurotensin expressing IPAC neurons were activated in mice who received the high-fat chow, but not those who had the regular chow, indicating that consuming palatable food specifically stimulated the cells. Then they did other experiments, and it's a nice little study in terms of trying to get a handle on what's going on here. But I do agree with you that with the brain, it's so complex. You affect one thing and you're not sure quite how you're affecting other parts of the brain or other parts.
You got to tackle it somehow, right? You got to go after it. Yep. And I think what the neuro, I feel like neurotensin is something we've actually measured at KCAS. I don't know why it's something we have like a biomarker, but
it's a peptide that we would do by LCMS. It's a relatively routine peptide for us to work on.
Yeah, And fascinating stuff, though, John. And certainly, like I said, I think I'd line up for that. I definitely I didn't have a lot of sweets growing like in my 20s and 30s, but I find myself as I'm getting older, it's like I got a sweet tooth, especially right around that 8 or 9 o'clock hour, which is the worst time to eat. So
Yeah, I was the other way around. As a kid, I was obsessed with sweets, but as an adult, not so much. Don't really bother me at all.
I was obsessed with pepperoni, meatballs, and pasta. All right.
We should not be talking about the food this early in the podcast.
Well, I got to mention pizza at some time, but anyway,
let's move on.
I think we got a couple more to go through. This is a good one.
We do, and they're all like really interesting things as well. So don't usually tend to talk about what specific companies are in, but are doing. Sorry, but I thought this was an interesting one that Eli Lilly commits $92 million. To Purdue University to establish pharma manufacturing scholarship program. As you know, drugs are getting more complex and delivery becomes more of an issue. Even going back 2 items ago, where we're talking about this cool synthetic enzyme, but we don't know how we're going to get it into the body. Here's Lilly investing in a local university to them and really looking to invest in Indiana to focus on this kind of activity. I mean, there's a research. So basically, there's money going towards establishing a scholarship program which will offer 75 to 100 students a year full tuition with guaranteed internship or co-op at the company, which is a huge opportunity for someone who's obviously Indiana based. But the key for me, and this is why I want to talk about it, is for the research part, the university will receive $50 million over the next 5 years with research focusing on genetic medicine, intrathecal delivery, and nanoparticle drug delivery. So it's actually forming that partnership to see how can we take these pieces forward, because this is where drug development is going. I thought it was fascinating that they're taking this approach to hopefully help their general research effort.
Yeah, I mean, it's really to help. They say Purdue University says they don't have a lot of homegrown Indiana students. I think that's a big part of this as well. And then there is a socioeconomic and educational challenges that this is supposed to, will help overcome in terms of just, getting STEM. These are all high schoolers that will be able to have an opportunity, 75 to 100, as you said, annually. Fantastic, John. I mean, I could comment more on it, but I'm going to be cognizant of time due to special guests. So maybe we'll do one last one between the two we have, and then we can jump right into the main portion of our.
So in that case, I'll jump into the one that I think we should do now, and the other one I'll hold till next time because I still think it's interesting, and that's this. In miraculous case, doctors treated a baby with a rare genetic disease before she was born. And obviously, we as a company deal a lot with other biotechs, etc., who are looking at treating rare disease, and obviously part of the challenge is, small populations, patients declining quickly. But one of the keys, particularly for pediatric rare disease, is treating early. And so for most, it's like you screen newborns, they're tested like blood pricks on their heel that then does we'll analyze various profiles. I know that Duke used to do the arrange of carnitines to look for various childhood illnesses, but if any of those analytical blood test results suggest the baby has a rare disease, then they can act on it. But this is one where researchers from UCSF, Duke, and the University of Ottawa treated a baby with Pompe disease, a debilitating genetic disease in which sugars build up in cells and cause heart and muscle problems long before. And the key to this is they did it before the girl was born. So it was before that typical newborn screen, and so they actually treated her when she was still in her mother's uterus. So they used an enzyme replacement therapy and other, and it was given basically to an unborn child. So the baby Isla was born last year to a Canadian family. Is now 16 months old. So far, she has met her developmental milestones and can roll around and sit by herself. At almost 1 year old, she began to walk by herself, and this is in stark contrast to two of her previous siblings who had one child who died at 5.5 and a half months old, and got enzyme replacement therapy early. The first one was diagnosed at 5.5 months old, but died at 2.5, and the second child was diagnosed prenatally, but died of 8 months of age. So the family I've dealt with obviously had a really bad time with this. So this actually gives them hope for this daughter.
Yeah, I mean, I had the, I guess, blessed or pleasure, or however you want to think of it, of having to work on Pompe disease 16 years ago. And so enzyme replacements and rare diseases are near and dear to my heart. And so to read this article, it's very uplifting. It's makes me just warm all around to think about how they treated in utero. That's an incredibly advancement there, and to have it to the point where her previous siblings didn't make it past 8 months, and this individual being 16 months of age and appearing to be somewhat developing accordingly. It's just why we got into science, John. It's an unbelievable scientific breakthrough. I think this is a unique. Pompe is pretty rare disease. Had the family for a third pregnancy. I mean, God bless them. That is, I can't imagine what that would be like.
Sure.
But it is something that Pompe disease is one of the worst forms of orphan diseases, in that it just they have a complete breakdown of the sugar. It can't break down.
Yep.
I mean, it is a wonderful article, and we at KCS treat we have several enzyme replacements themselves, as well as that's where gene therapies and cellular therapies are moving, as well as into the the rare diseases, and kind of knocking in enzymes using cells rather than just recombinant enzymes themselves, which you could argue. I could share why they're the pluses and minuses of routine recombinant versions versus a genetic version. And the biggest one is you got to get monthly or quarterly treatments versus a one-time shot with the gene therapy. So
sure. So that does play into it. So Priya Kishnani, who was one of the lead authors on this, said that the intrauterine treatment was not a functional cure, but at least it bought the babies time where hopefully there'll be other one-time treatments such as gene therapy that can hopefully cure them for the long term. But equally, I mean, at least it's a start that's given the baby at least a chance of a normal upbringing, and obviously they'll be monitoring in the long term to just observe how she's doing. There's actually they actually have more children enrolled in the study. One with Hunter syndrome, and then another one with Hurler syndrome, and both those are doing well. So they're obviously this was the infant we cited as the first one, but there's others are going through the same approach. So, I mean, it hopefully it could be a very powerful approach for the long term. And I just thought it was a really enthralling piece of research.
Yeah, I want we'll move on to our main topic here, but it seems like it has to treat in utero just sets the child and the adult up for success, and well, at least I say success, but for health.
Sure.
Because, they're already addressing and you wonder how it may help with immunogenicity is where I was going, John, in terms of, is this something that, where they're getting exposed to the enzyme in utero, will that help? Because a lot of enzyme replacements, they start to fail because of immunogenicity.
Okay.
Genetically, there's even, that's putatively happening as well, in terms of you start to develop antibodies that will actually bind to the therapy or the therapeutic cells that are engraft. So I digress, but it is something that I, that kind of where my mind goes. I wonder if this will help the child throughout and the individual throughout their life with being treated at such an early age. That's exposure, right?
Sure. Yeah.
That's how you build up immunity. So without further ado, John, I think we should jump right into our main portion. We have a
let's go for
very
it.
exciting guest. We've got two special guests. John, why don't you tell us about our guests?
I was going to let you do it, but okay, yes. So the main topic today is introduction to active biomarkers, and we've special guests, John Bucksath, who's our CEO, and Marc Essodaigui, who is the general manager at Active Biomarkers. And so I think probably, well, first of all, welcome, gentlemen. If you want to introduce yourselves, John, I know you've been on the podcast before. At least if you want to say hello, and then we'll pass it on to Marc and get the conversation going about Active.
Absolutely, John. Thank you for having me on the podcast again. It's a pleasure to be here. And just a quick intro of John Bucksath. I've been with KCAS as CEO a little over 6.5 years to this point, and it's just been an absolute pleasure to work with this team and see the progress of the company continue to accelerate and grow to really be a standout CRO providing development for the bioanalytical biomarker arena for worldwide now. And we look forward to talking that with Marc here just shortly.
Marc, it's our pleasure to have you on the show here. And why don't you introduce yourself?
Well, thank you very much. And thank you for having me here. It's a great pleasure to be with you. Thanks for the invitation. So very briefly about myself. Well, I'm 53, father of 3. I'm French. You can hear it from my accent. I am indeed the general manager of Active Biomarkers, a Lyon-based company. I've been in this position for almost 7 years now. And before that, I was taking care of the business development of the company. My background is more in sciences, and especially molecular biophysics. And I was carefully listening to your discussion regarding those, breaking news. And I'm always impressed how fascinating this world we are living and working in is. Exciting as long as it impacts our lives every day and for everyone. I've been working in the so-called field of personalized medicine or precision medicine for more than 25 years now, notably in the life sciences industry. And I have had the opportunity to work with great teams and focusing on several diseases such as cancer, including in my PhD, but also infectious diseases and neurology, just to name a few. So I'm really passionate about science, but also how it is impacting our lives. And I see it also as a great opportunity for business development. And this is what we try to do at Active Biomarkers. So that was for myself. And again, thank you for having me here.
Yeah, welcome, Marc and John, and we're excited to dig in a little bit. This is an open forum, so we can take this however we like. But I think the first thing that our audience may want to know more about is just maybe a little history of active biomarkers. And Marc, I'll turn to you to kind of tell us a little bit about the history of active biomarkers, and then we'll jump into the path to acquisition.
Well, thank you. Sure, definitely. So Active Biomarkers was founded in 2011. It has had a very original birth because it has been founded by 2 French biotech companies developing immunotherapies in cancer and infectious diseases. And to name them, these are Transgene, a company located in Strasbourg, close to Switzerland and Germany, and another company called Innate Pharma, located in Marseille. And at Innate Pharma, they are specialized in NK cells, and they leverage the immune system to fight cancer. So none of them was happy with their potential in finding a partner to support their immunomonitoring studies and biomarker studies for their clinical trials. And because they were unsatisfied, they decided to create that company, formerly Platin Pharma Services. And this is how it all started. So pretty original, because the first foot was or first step was directly into the clinic, which is not that frequent in our market. And it started with the team of 14 people. Half of them were former employees of Innate Pharma, and the other half was from Transgene. And I have been the first alien, so to speak, in charge of the business development. This is how it all started. It's an 11-year story that has been extremely rich in terms of developments. In 2013, the company was acquired by a US CDMO called ABL, located in close to Washington, D.C. area, and which is an affiliate of a renowned group called Institut Mérieux that is focused on infectious diseases. And this part of our history helped the company develop itself in additional fields such as CNS and autoimmunity. What we do basically at Active Biomarkers is really giving the samples or the biological samples the opportunity to give us some actionable information. And we do that for all our clients in the biotech and pharmaceutical industry, mostly in Europe, in those indications that I have mentioned. So the technologies that we have in-house encompass a broad range of immunoassay technologies, starting from the ELISA to some multiplexing technologies such as MSD or Luminex, or even ultra-sensitive techniques such as single-X historically, and now the Simoa technology from Quanterix. The other pillar of the company relies on our know-how on cell-based assays, and notably flow cytometry and ELISpots. The company accounts almost 35 employees currently, and we operate in brand new facilities, all BSL2, and we are, of course, ISO certified, GCLP accredited, and GLP compliant. So just to give you some background information about Active Biomarker history.
No, thanks. I think your founding in 2011. That's right around. I had been in the CRO space for a year or two. It makes me think about how when I joined the CRO space, people said, "Why are you doing that? How come you're not going into biotech?" Right? Because that seemed to be at the time it was thought of as like bottom feeders. Although now it's completely reversed, and maybe we'll have you on another time to talk about that. I'm sure you've experienced some of that. But now, CROs are really I think, a critical part of most companies' success. So a fascinating story there. I'm learning a lot about active biomarkers myself. You talked about a lot of platforms that are near and dear to my heart, but maybe we'll turn to John Bucksath and maybe tell us what your vision, John, I don't know where I think of it as a vision, but I'm sure when you engage them, maybe tell us a little bit about how it all came to fruition in terms of the acquisition.
Well, sure, Don. With your comment there about bottom feeders, that's it's funny how industry has evolved over the years, where outsourcing first started out as I refer to as more of a pair of hands type of approach, and today the CRO industry suddenly disappeared. There'd be a complete collapse of the R&D engines and ability to get products through the development phase. So what an evolution the CROs have taken in the support of drug development. That said, obviously, the expectations of a CRO today are much, much, much different than they were from 2011 and beyond. Historically, again, going from a pair of hands to today, arguably much of the expertise, particularly within that CRO's expertise or what the services they provide, arguably are, at least on average, for the actual work that they perform, that's where most of the expertise resides in the industry today. And of course, I'm biased probably in that perspective, but the reality is, when you think about it, the CROs today see more projects, see more studies through their various clients, and arguably would make the melting pot, if you will, of best practices on how to accelerate drug development efficiently just through the continued practices. Obviously, that also has played out in how the market has evolved. For us, obviously, KCS starting out as a focused bioanalytical provider, as we've grown, as you call it, Dominic, the vision, if you will, was to continue to support the development of our clients. And of course, like any business, we want to create value for our investors and at the same time focus on the patient first, if you will, and really understand that we provide an incredible service in helping these life-saving, life-changing drugs to get to the market. And that's what gets me up out of bed in the morning when you think about those type of opportunities and what impact we have on the world. As we continue to grow the opportunities and to really understand the shifts in drug development, really from what apparently was, particularly when I was in the laboratories, more of a country-focused type aspect. Drugs were more commonly developed, for example, in the U.S., and then that country would decide, okay, maybe now we want to think about moving that into a European theater and also expand our market. Today, with the ability to move data, more of a global reach, that has really morphed into evaluating all of that market at once. And as we all know, the criticality of development, particularly around the clinic aspects and finding these patients, if you will, that can participate in these trials, really opens up the bandwidth to speed up some of these trials as the trials ever more complex and more challenging. It was just yet another reason to provide and look globally for continued drug development. Of course, when that happens, then as these companies look to be more global, they want their partners to be more global. And we knew early on at KCS that with our vision where we wanted to go as a company, that we were going to have to consider how we were going to meet that need for our clients as that continued to evolve. From there, you really have two choices: either go, do it greenfield and build it yourself, but more importantly, in which most people want to do, or most companies tend to do, is try to find an organization that they can partner with, and that have the expertise, that have the leadership in place, that can really partner in the old approach of 1 + 1 = 3 to really support the clients to move into that next level, and quite frankly, open up the market too for the CRO itself and continue to expand. Period. It's really not for the faint of heart. There's a lot of challenges to that. Obviously, just something as simple as the geography, the time zones, all those things that we take as just normal. But there's also the aspects of different cultures. So really focusing in on finding that right partner with the leadership, obviously the talent was key for us. KCS had been looking for several months, well over a year, for the right partner. We actually looked at other organizations before we were able to, get Marc's and his team's attention, so to speak, and quite frankly, turned them down. Some arguably much larger, but we were looking for the right organization. Again, they had the expertise, had the commitment, had a similar type focus of growth, expertise, obviously leading with a quality product, quality data provided to the clients to help them, again, get their drugs to market. So we were absolutely thrilled when Marc was at least open to discussions, and over quite a period of time, we came to know each other. While you want to move forward in the business, it was absolutely critical that we get it right. And today, I'm very thrilled to say that Marc and the team is partnering with the teams here in the U.S., and we look forward to continue to evaluate how we can come together. And we've already seen some opportunities from our clients that we currently have that are very interested in hearing more about active biomarkers. And we also know that some of active biomarkers customers previous to the KCS acquisition now are interested in hearing more about, how we can come together. The beauty of it is, they have significant expertise throughout their platforms. But like any organization, you tend to have more strengths in some areas. So what we're finding is that Marc and his team has really been key to us really thinking more about some of the work that they do, the expertise that they bring that really, really shores up some of the areas that we were hoping to evolve here in the U.S. And then I think we're able to provide here in the U.S. Some expertise to support Marc and his team. So that's really when you start to see those synergies come together and you see the teams working together, it's a really exciting time, quite frankly, and what we can do and move into that platform of being a global provider, a focused bioanalytical and biomarker provider, servicing all aspects of any type of development across the range of molecules today, everything from NCEs to biologics to cell and gene therapies. And that really is our focus. And again, we see incredible opportunities in the market. And but that first and foremost has to start with the foundation of the talent, the people, and the leadership. And again, I can't stress enough how thrilled we are to have Marc leading that, leading the European theater for us at this time.
Yeah, no, John, as I sit here, I'm thinking 6.5 years we've been together, and this is it's been a fun ride with you. And I'm getting to know Marc, and I'm going to let Marc kind of give his thoughts on the path to acquisition. But I knew from day one when Marc spoke to our entire company, I said, "Oh, I can see why John likes him." So, Marc, maybe take a few minutes and tell us your thoughts, and then I think we can really dive into some of what the acquisition means for KCAS and Active. But maybe share a few thoughts on, building on what John said through this sort of wonderful journey that brought our two companies together.
Sure, definitely. But before answering that specific question, I would like possibly to add just a very brief comment to what John has said. And thank you very much, John, for your kind words. Clearly, I would like to add that on top of everything that can seem absolutely logical because of the complementarity of the teams and the projects, I would say that since day one, the discussions have been extremely fluid because despite those differences in cultures, in time zones, in languages, and so and personally, I immediately also felt that we were sharing the same vision, the same care for quality, scientific, quality, and these are absolutely central pieces for our employees, for our clients, but also for me and for the scientists that we are all, to make sure that we are contributing to something and improving global health at our scale, no matter, how small it can be. We are contributors there. And that, common vision has been really absolutely a cornerstone to making that discussion, eventually successful. So it's because we were sharing the same vision that we have completed this operation acquisition, and that we have decided to join forces. Now, regarding the path in itself, as John mentioned, it's almost 1 year of discussion, very thorough due diligence to really get to know each other. Very frequent meetings, but in the end, plenty of logics, as I mentioned. It can be, from our respective experiences and expertise. Say, in small molecules, we don't have a huge experience at active biomarkers in small molecules, but we do have in large molecules, biologics. It can be bispecific antibodies. It can be antibody drug conjugate. It can be cell therapies, CAR T cells, vaccines. We have worked so much on very innovative vaccine, anti-COVID vaccines, anti-flu vaccines, and so complementarity of types of therapies, complementarity in technologies. We know very little about mass spectrometry as active biomarkers currently, but we are bringing a very large platform of immunoassays, as I mentioned earlier. So complementarity of techniques for our clients, because sometimes, when you want to develop an assay, you will try this approach, but it may not always succeed. And now, altogether, we can offer a much wider spectrum of solutions in order to make sure that we have explored a maximum of things in order to bring the right solution to our clients. Complementarity of geographies, for us, that's absolutely obvious. We have supported at least 100 clients so far, more than 600 distinct projects. Most of them are phase 1 and phase 2. When we want to get into phase 3, we needed to have, the right partner, as John said. Multicentric studies are absolutely central. The United States and Europe are currently, the places where most of the clinical studies are conducted. Being present there, offering that proximity, helping our clients also deal with logistics, sample fragility. This is absolutely key. And last but not least, of course, and you gentlemen here are just the perfect example, bringing also top level scientists together to bring our clients the best solution is also a great driver for having joint forces. So, long story short, plenty of complementarities. So it was, I would say, a natural move for both companies.
Yeah, if I can jump in there, at least on the small molecule piece. I mean, we've talked to your team about where we can help, and certainly there's clients that you're working with who are looking for small molecule biomarker support that we have expertise in, and yeah, we look forward to spending more time working with you and hopefully training your team to learn more about mass spectrometry as well as to where it can help you to complement your existing technologies.
Yeah, I'll just add that we have an immediate opportunity, right, John, that we have a call coming up, I think, tomorrow with that.
Sure.
This is the, why the two organizations have partnered and merged is that the very thing you were touching on, Marc. It's, all of your expertise and heavily involved in what I will say ligand binding assays. We complement that over here in the States. So it's really it's kind of a twofold thing in my thought process is we bring that mass spec component of things to you, but then it really does open us up for the ability to support you said it, larger global studies, and that's something that we're still I'm working with Magali, I hope I'm saying her name right, and she's been wonderful to work with, and we have a couple irons in the fire there, but as we start to integrate more, I anticipate we'll have more and more opportunities. But we've had a few out of the gate, and excited to. And when I say what I mean by that is, you're running samples in Europe, we're running the same samples here in the U.S. We have a lot of complementary equipment. The future is very, very bright. It's incredibly exciting to finally, kind of hit the ground running. And as John mentioned, we do believe we'll have our first true. Where you're going to be running a sample in Europe and then shipping that sample to us, and we'll run it on a mass spec here for biomarker analysis. And that's really, I believe that's the vision of both of you, John and Marc, in terms of when you thought about what the acquisition would look like going forward. So with that, I mean, that leads right into our next topic, which is the collaboration between KCS and Active. And I don't know, Marc or John, which one of you wants to go first, and just maybe we'll talk a little bit more about, I touched on it a little bit, but maybe you can elaborate on how do you see more of the future of how we'll collaborate?
John, you want to start maybe?
Sure, I'll jump in on that. And, obviously the vision, as we've been discussing, is to really serve that market that is looking for global, not only just regionally in, the European theater and the U.S. theater, but also, again, across the globe as we continue to move forward. We are still open to, potential acquisitions that fit our paradigm of being that, expert provider across the platforms. We're actually in the market, if you will, with looking for opportunities, whether that's through organic means or acquisitive means, to add mass spec in Europe. And I know Marc is actively looking at some opportunities there representing the company in that regard. So we're still very open to that. So I think that definitely checks the box as collaborative. Again, having Marc's leadership there has been key for us to have a lot of confidence and to be, for lack of a better word, more, I think the poor word maybe is aggressive, but at least knowing that we have the talent that can take on the opportunities to continue to serve our clients across those platforms. With those things in mind, we're going to be choosy about how we do that. But again, we do think that having very similar profiles on both sides really allows our commercial team to really work with the clients and understand their needs. And I think that's a pretty strong checkbox, if you will, for a lot of these global needs for a lot of these providers, for a lot of our clients that are looking for, that synergy, right? If I have samples coming out of Spain and I want them to be the same as some of the same clinical samples that are coming in the US, and I can send them to an organization and speak with, a team that has all of this together, and they can share the results between these different geographies in a platform, in a form, in the reporting of the data in the way that I've agreed to. It just makes their life so much simpler. And, that really is starting to limit the number of CROs that can do that. And with significant amount of consolidation in the CRO industry over the last 5 years, that demand, if you will, from the industry is only increasing. As we look at it, the percentage of compounds in development have gone up, but as we start to look at the clinical development, as those compounds, theoretically, let's just for argument's sake say that the number of successful candidates moving through the non-clinical aspects hitting the clinic, well, the reality is there's way more molecules in the pipeline today. Now multiply that time, this big shift for compounds not only to be regional but global. There really is a big shift in looking for those CRO providers that can service globally. And so that's exactly where we're positioning it. So the collaboration there is to really, as much as we possibly can, bring our systems together, get on the same platforms. Obviously, there'll still be differences to meet the regulations and requirements, for the regions as well. But right now, that's probably one of our biggest folks as a combined organization as to what do we need to do next? What systems, what investments do we need to make so that we can go to our clients and say, look, we can help you develop drugs globally. And here's why. Here's our systems. Here's our quality management systems. Here's our technical platforms. Here's our talent. Here's how we communicate. Here's how we can help you as a service provider to make these things happen. Conceptually, if you will, using the word vision again, that's easy to say. Very challenging to pull all the pieces together and make that work. But again, that's why we were so picky, and again, thrilled to find Active Biomarkers as an opportunity to partner with, and then move the organization to position the organization strategically to be able to do that and compete in that market. And we're well on our way to doing that. So a lot of exciting news coming down the way here in the next, I won't say several months. Several months might not be that many, but here in 2023, we'll have some exciting news, and I won't preempt that here on the podcast, but definitely we'll have more fodder for discussion as the organizations continue to work together to create a solution for the industry.
Teasing us a little bit there, John.
He's angling to be a more regular guest at doing things.
Hey.
So Marc, maybe you want to add anything, and then we can maybe go to the next topic.
Yeah, very little to add. It's really the people tend to call it integration. For active biomarkers from our side, it's really a great opportunity to unify our processes. We may act locally differently, but clearly we as a company are learning a lot, and we are impressed also by the level of structuring of KCAS at every level of the company. So we organized in work streams, and I can already see the results in several departments, say marketing, sales and business development, quality assurance, information technologies. At every single level, we can already see the change. Of course, it's a work in progress, as John mentioned, but a lot of excitement and a great future.
Yeah, I'm sure you're getting a full dose of forecasting and scheduling and planning as well, which are near and dear to John Perkins and I heart, where we help champion some of that. And it's one of the, I guess, really John brought that when he joined KCAS, and it's been a wonderful experience for me personally to learn all about that.
So we are in that learning phase,
yeah,
and it's great. I can confirm.
Yeah, I mean, when people say they're process driven, sometimes it might not mean anything. But when we say it at KCAS, I think, Marc, you're feeling that, like from the moment that contract is put together to the moment it's executed upon, and then to the moment that it's billed, all of that has a very specific process. And John has brought those, really the three tools here. It's forecasting, scheduling, and what we call our CURE model, which is actually, tells us it's an accounting type of tool. But it's been a great ride, and stay tuned, Marc. We'll get to know you a lot more on Tuesdays, right? That's when we have our forecasting. So, I don't know, John Perkins. I feel like I'm dominating a little bit here. Did you want to maybe jump in, or I don't know? We can certainly follow our agenda if that's.
I think that just go with the agenda. I'm fine with it. I mean, we're conversation's going well, so I'm good with it how we are.
Yeah. The next item we had was just, what does the acquisition mean for existing customers? And I don't know which one of you wants to jump on that one first. I know that from our perspective, we have a lot of our customers that are still in that, you got this site in France and we have interest in it, but maybe just talk a little bit about what it means for existing customers, and then we'll talk about future customers.
And I will have something to add about that, but let's let John and Marc go first.
Okay, well, I'll jump in just with real world observations so far. I touched on it briefly in that opening salvo, but just hearing the clients say, "Wow, okay, so you have additional capacity, backup, you have." A larger talent pool, if you will, so you can call on for additional help, going both ways. So, from the client's perspective, when you think about it, they're in a very risky business, right? And so the one thing that they don't want, the success of the drug itself is very risky. What they don't want to also have to address is my provider a major risk? So more capacity, more expertise, those type of things, at least fundamentally on the paper, if you will, appeal to these organizations when they realize, hey, they have the capacity, expertise, they've added to that, but also their reach and understanding and ability to service our needs as we continue to develop drugs. So we've had several inquire clients that we're working with in the U.S. now have said, okay, we understand what you do. You guys do a great job. This is wonderful. This is the range of opportunities that we have under your current, offerings. What's been interesting to hear from the commercial team is now talking to those same clients who maybe have very large portfolios, use multiple CROs, are interested in inviting us back in to have different discussions. Okay, what's this going to look like in the future? What can you provide? And from there, again, signaling a need that there's just much fewer CROs out there that can really provide that type of global reach. And as we continue to integrate, there's just several clients that want to hear, when we believe we are ready, and that's critical. We don't want to go to them and say, hey, we just popped the champagne cork. We're working together. We're partners now. We're mature enough to realize that there's a lot of work to do to get our systems together, to work together before we go approach these clients and say, we're ready to take on some of your global programs. But that being said, the the opportunity to really work with these clients and ask them, what's some of your biggest headaches? So like, well, I work with CRO A and CRO B and CRO C to meet these needs. And we just simply don't see providers out there that can service those needs. And so immediately, as you can imagine, dealing with three entities, it'd be like going to get your car repaired and you have to take it to three different repair shops to get the whole thing repaired. So with that in mind, obviously, if you got one shop you trust, they do good work, that's going to be a much less stressful and arguably less risk for continued development services being provided by that CRO. So ultimately, we're hearing, hey, clients are like, great, that's fantastic news. We want to hear more, but we also want to make sure that you guys are ready. And I think that's the key. Some CROs that have made acquisitions before, it's been more of a, quite frankly, let's do a roll-up aspect and slap a brand on it. And then you're really operating as individual sites, and they're not really what I would call, technically integrated aspects. And that, while that might get some of the work done, it really does not position that organization to grow and grow with the industry. So we're very focused again on getting those systems together, and that's not asking active biomarkers to adopt everything that is what's going on here in the States. It's mutual on what is the best process we can come up with that serves both the needs, and that's been a very eye-opening scenario as Marc and his team have really opened the U.S. team's eyes on a lot of processes that they run that are actually quite much more efficient than what we do here in the U.S. So really excited about taking advantage of those. But in a nutshell, I think that's that collaboration aspect that you've asked, Dominic. I think for me, that's what is just really validating what we believed was the opportunity in the market. And so far, the responses have been quite positive. So really looking forward to, getting out there, talking to the clients, learning more, and say, here's how we can help. And of course, we'll have a much clearer picture as those things evolve.
Yeah, I just know when I'm in a situation where people are getting to know the KCS brand, it's very powerful to be able to say we have now really 4 sites, but specifically a second, really one counter to us in the LBA space, specifically in Lyon. And it's people have definitely view us much differently now if they hadn't heard of us versus just being a single entity in the U.S. and now having multiple entities across the globe and growing, right? Marc, did you want to add anything?
Yeah, very little. Going back to the fundamentals again, difficult to add much to what John shared with us. But what I would like to say is finally the philosophy. When you look at drug development, it's a matter of of course science, but also risk management. The stakes are high, and in the end, when you look at companies like, let's mention them separately, KCAS and Active Biomarkers, what are we doing? We are really concentrating talent, knowledge, technologies, of course, in order for those clients to avoid, useless expenses, time that it takes, talents to recruit, and so we are concentrating just that. So when we decided to join forces and what it means for the future for those clients, existing ones and new ones, it's really that ability to put our know-how, expertise, which is experience multiplied by skills for me, and bring it all together and across several types of therapeutic approaches. And I think this is really unique with that objective of really making those synergies work. And you clearly mentioned it, and Magali is the right name. You said it well. It's already working. We have already several of our clients in direct contact with our new US colleagues to address some complementary questions that they had. And that's exactly why we are, developing this one company. again, the added value is that concentration of experience in order to save time, resources, and really mitigate those risks that as much as possible for our clients, in order to avoid all the useless mistakes that they may make during their development. So that's really the promise of this business and for our companies.
Yeah, and we're running a little bit short of time.
I'm just going to say, Dominic, sorry to interrupt, but obviously I'm just back in the US after spending some time in Europe, particularly at the EBF meeting in Barcelona, and we had a booth that was presenting both KCAS and Active as well as Flowmetric as three companies under one collaboration, and there was genuine excitement with customers at the booth and finding out who we were and possibly how we could help them. And it's one of the trade shows I've done where I actually came away feeling that this it was really positive feedback from potential customers, and really showed the potential of us and Active working together.
Yeah, I think I'll add, Marc.
Sorry, it made me think of something I completely forgot to mention. It's the facilities. Our business has that unicity. You may start a virtual biotech with 2 or 3 people working in offices with tens of vendors, and you can develop your drug. If you want to do our business, you need to invest strongly into, state-of-the-art facilities, BSL2, sometimes BSL3. And this is also something that we have in common. We have relocated our labs to double our surface in 2021. KCAS has magnificent facilities in Olathe. I never know how to say it. Sorry, but close to Kansas City, with 7,000 square meters of really brand new labs that really impressive. And not to forget, of course, our colleagues at Flowmetric that are about to relocate into a brand new site. And these investments are very important. So for our clients, this is also a great, gift that we are bringing together with our services and know-how.
Yeah, absolutely. It is important. The facility is, anybody who wants to come to Olathe, it is just outside of Kansas City. We'd love to host you, as well as certainly, I don't know if I'll make a trip over to Lyon, but I'd love to see your site.
Certainly, if you're interested in the Lyon facility, there's a small tour on Active Biomarkers account on LinkedIn, so you can see that to get a sense of the labs there.
Yeah, we did it. We have a great marketing team, and they did a kind of teaser. So you can only see the first part. These are the offices. And if you're patient, you will probably see soon the labs. But we are offering a kind of virtual tour to see how and where we work.
Yeah, and we're running right up against the time here, but I'll end with one last thought for me is, when we think of our future customers, one of the really powerful things about our merger is, Marc, you talked about most of it was in phase 1 and phase 2 biomarker analysis, and we're in a similar place. We do a few phase 3, but I think a lot of clients, when they hear they're in the phase 1 market or the phase 2 area, they're always looking future-wise for their vendor or their CRO and thinking, could they support my phase 3 and could they support it globally? And now collectively we can answer with an affirmative yes, right? That's a very powerful, for me, I don't want to say selling point. It's just, it's a very powerful future for both of our organizations to be able to leverage both of our talents, facilities, and expertise across the globe here. So I'll leave with that. I think that's to me, it's something that for my 9+ years working here, we'd always had to kind of dance around well, can you support phase 3 and do you have a European footprint? And all of that now is yes for us. So I hope that's the same on the biomarker side, the active biomarker team.
clearly, I couldn't say it any better. So yeah, perfectly. And we have a number of examples of very happy clients that had to leave us, so to speak, for their phase 3 because we were not able to support them globally. And now the answer is yes.
Yeah. So I'm getting a message that we've got to wrap this up. Gentlemen, you're going to avoid the hot seat. The hot seat is one of our, well, I call it the hot seat. That's John and I like to ask you a few personal questions, but we're really out of time. But it's been a pleasure to host both of you. We welcome you to come back. Maybe we'll have you back in a few months or whenever you'd like, but you're always welcome. And I'll let you maybe give some parting words, and then we're going to have to wrap it up.
Well, I just say thank you for having me on the podcast, and it's been a joy, and obviously shoulder to shoulder here with Marc, and moving forward, I'm just thrilled to be able to share a little bit more information about our combined entities.
And for me, merci beaucoup. It's been a pleasure being with you today.
Yes, yeah. Thank you very much for joining us.